speaker is Stefan Demotz.
So thank you for the introductions. I'm Stefan Demotz, and I'm going to tell you in the next 15 to 20 minutes about the work we are doing for the development of a drug candidate for the potential treatment of GM1 gangliosidosis and Morquio disease type B. First of all, before I discuss the development of this molecule, I will present two slides about the company which is developing this molecule, because I think it's interesting to understand the context of this work. So the whole story was started 7 years ago when a couple discovered their daughter was afflicted by Sanfilippo disease, which is another lysosomal storage disease, very rare, orphan. So their reaction at that time was to create a foundation. And I was involved in right at the beginning in the scientific committee of this foundation, and we had the opportunity to finance research projects. They developed pretty well, and we reached a point where it was no longer to push further these projects as a foundation, so it's why we created a company for logistics, commercial, legal reasons. And since the company is owned by the foundation, the objectives of the company are aligned with the objective of the foundation, which is the development of treatment for lysosomal storage diseases, mucopolysaccharidosis in particular, and related diseases like GM1 gangliosidosis. So this is a very small company. It's asset-centric, meaning that there is only one product in development. We are 2 employees. And since we are so small, we outsource most of our activities to academic labs, to service providers in Switzerland, in Europe, and also in the States. So we are busy organizing all these collaborations, and we have also a small lab at the University of Lausanne, and we work also in the lab. So there are different ways, as we'll see this afternoon, to envisage the development of a treatment for GM1 gangliosidosis, stem cell transplantation, gene therapy, enzyme replacement therapy, substrate reduction therapy, and the pharmacological chaperones. So this last option, it's the one we have chosen, and we are trying to develop. So for those who are not familiar with the pharmacological chaperones, here is a schematic representation of the mechanism of action of these pharmacological chaperones. So when everything goes well, lysosomal enzymes like the beta-galactosidase folds properly after synthesis in the endoplasmic reticulum, and then moves to the lysosomes where the enzymes do their job, which is to degrade cell constituents like GM1 gangliosides. In the disease situation, the protein is mutated. It does not fold properly, so it's recognized as faulty by a cell quality system, so it's immediately degraded. So it doesn't reach, of course, the lysosomes, and there is accumulation of cell constituents which ultimately lead to the disease. So the idea of the chaperone compounds is to use a small molecule which tightly binds to the mutated protein, so it helps its folding, so it looks normal, so it's allowed to leave the endoplasmic reticulum and to reach the lysosomes where the protein now can do its job to degrade cell constituents. So the small molecules which tightly bind to protein, to enzymes, are often inhibitors. So we use a paradoxical situation where we use an inhibitor to restore the beta-galactosidase activity. The beauty of the thing is that in the acidic environment, the compound is released from the beta-galactosidase. So chaperone molecules, next slide please, offer advantages over other therapies. Since they are small molecules, they pass the blood-brain barrier. It's very important to reach the brain since it's a target organ in the disease. They are small molecules. They don't cause immune reactions like it's often the case for enzyme replacement therapy. They are also orally available, so it also makes the administration easier, especially as compared with enzyme replacement therapy. Small molecules are also cheaper to produce, more or less. A drawback is that they usually work only on a fraction of the whole array of mutations. In our case, as we'll see in a minute, the compound is capable of restoring part of the beta-galactosidase activity in about 2/3 of the cases. And this is, in a way, comparable to Amigal, which is being launched these days by Amicus Therapeutics for the treatment of Fabry disease. It's also a chaperone compound, also an immunosugar-based compound, and in their case, it restores some activity in 60% of the cases. And this launch of this chaperone compound by Amicus Therapeutics, it's also good for our development because it's a validation of this approach. They have successfully gone through the clinical development. So this story was started end of 2011 at the time of the Swiss San Filippo Foundation. I established a collaboration with a chemist at the University of Orléans in France, and he started to develop, to make medicinal chemistry around this molecule, which is a moderate but not so specific inhibitor of beta-galactosidase. And after 3 years of work, we ended up with this compound, as we'll see, is a potent and specific chaperone compound for the human beta-galactosidase. So these molecules, so it's a potent and specific inhibitor. It tightly binds to human beta-galactosidase, and this is illustrated on the next slide. Here you see the cycle in the catalytic site of beta-galactosidase and with this alkyl chain protruding out of the protein. So it illustrates there is a tight binding. So now the question is this compound capable of restoring beta-galactosidase activity in patient cells and also to decrease accumulation of cell substrate? And this is these two questions are illustrated on the next slide. So in patient cells, there is low activity of beta-galactosidase, low activity of beta-galactosidase due to degradation, accumulation of substrate in the lysosomes. So now when we treat these cells with a compound, do we restore beta-galactosidase activity and do we decrease accumulation of substrate? So the answer to these two questions appear to be yes. So first of all, here, first piece of results, we have taken cells from a GM1 gangliosidosis patient, and we have treated these cells with increasing concentration of compound, as indicated by these values. And you see, and after 5 days, we have taken the cells and measured beta-galactosidase activity, and you see a gradual increase in this restored beta-galactosidase activity, starting from a low activity up to reaching a plateau. And there is no difference in another lysosomal enzyme, beta-exosaminidase, which in a way proves the specificity of the activity. So we have repeated this test now on over 50 patient cell lines and found that in 2/3 of the cases there is possibility of restoring at least part of the beta-galactosidase activity. The next step was to show that this treatment results in a decrease in accumulation of the substrate of the beta-galactosidase. So there is 2 substrates for beta-galactosidase, which is GM1 ganglioside and keratan sulfate. So we use this second one, which is an assay which is not so easy to run. So in cells from a healthy person, we measure keratan sulfate, a little bit of keratan sulfate, 18 nanogram per ml, and this is close to the limit of detection. So it means that there is very little in these cells. When we take cells from a GM1 gangliosidosis patient, we detect a lot more, 10 to 12 or more. when we take these cells and we treat them for 1 week with the compound, then we measure the level of keratan sulfate in these cells, we have a decrease of 30 to 40%. So this establishes that this treatment is capable of reducing the load in one of the 2 substrates of beta-galactosidase. And this was the goal of the study. Now you can say 30–40% reduction is not that great. This was not a test which was optimized in terms of dose, of duration of the study. It's very expensive. We had not the luxury to repeat and optimize this test. The goal of showing this efficacy was obtained. So it's about where we are. On the next slide, this is a list of activities which have to be done before to be ready to reach the phase, the step where we can start to think about the clinical trial. For instance, I can mention a few. We have to measure how the compound disappears in animals, in a living organism, to show that it distributes in the target organs, which are the brain and the bones for Morquio disease. We have also ideally to show that it's therapeutically active in an animal model of the disease, to try to devise how we can administer the compound, and also to show its toxicity, to evaluate how toxic it is. So to conclude, I would like to present 2 pieces of results to illustrate that we have started to work on these aspects. First of all, we have started to evaluate whether the compound goes into the target organs. So we have started to work with mice and rats, and when we administer the mice either intravenously or by oral administration, and then after different period of times, we take groups of animals, they are sacrificed, and we determine the level of compound in the brain, in the plasma, and in the bones, which are the target organs of the disease. And on the next slide, you see the level of compound following 3 intravenous administration, 1 hour after the last one. So we detect quite a bit of compound in the plasma, even more in the brain, and even more in the bones. So this is good news in the sense that the compound reaches the target organs, and we can reach concentrations which are well above the concentration needed to obtain an effect on cells of patients in vitro. Should mention, this is not yet optimized, far from that. A last piece of result is related about the identification of the responder patients. So as mentioned at the beginning, a drawback of the pharmacological chaperone is that they don't work on all mutations. There are some mutations are not responsive. So it's very important to be able to detect, to identify the patients who bear mutations which are responsive to the compound. So we have started to devise an assay which we wanted to be non-invasive and rapid in order to identify the right patients. And this is illustrated on the next slide. So we take a little bit of blood. It can be done with less than 10 ml of blood. We isolate from this sample of blood the leukocytes, so the white cells, and we expand these cells to obtain B lymphocytes using a virus, or expanding the T lymphocytes using some reagents, and this is done in the presence or in the absence of compound. And then after a few days, we measure the beta-galactosidase activity in these cells. And we have applied this assay on a sample of blood obtained from a Morquio disease type B patient. Since she is an adult person, it's also easier to do this test on from blood sample from an adult than a child, obviously. So here in blue is the activity of the beta-galactosidase in cells expanded from the blood sample in the absence of compound, and here in the presence of compound. So you see this substantial increase in activity, which illustrates that this patient bears mutations which are responsive to the compound. So this person is potentially a subject for a clinical trial. So in summary, we are in the process of developing a potent and specific compound which works on a large proportion of mutations of the beta-galactosidase, about 2/3. It has characteristics which make it easily handleable. It's water-soluble, it's non-cytotoxic, it's orally available, and it penetrates the bone and the brain, which are the two target organs. An aspect which is often neglected, we have been able to scale up its manufacturing. It's something to produce a few milligrams. It's another story to produce gram quantities or even a kilogram quantity. It's quite challenging from a chemistry point of view. And we have also devised a synthesis which is suitable to produce the compound in a way it can be administered to humans. It's also something which is not trivial. When we compare the features of this compound with Galafol, so the compound which is being launched by Amicus Therapeutics for Fabry disease, the features are very similar. And since this compound, Galafol, has made it through the clinical development, it's also very encouraging for us. It's a precedent that we would like to follow. And finally, I've briefly presented this assay to identify the patients who are responders. So what are the next steps? So as a summary, this story, this adventure was started by the Swiss San Filippo Foundation. The discovery phase was done by the foundation. Then the project was handed over to Dorfen. We went through the non-regulatory preclinical phase. This is about the data I've briefly presented today. Now we have to go through the regulatory preclinical phase, so it means toxicology, GMP manufacturing, and also the setup of the clinical trial. And then there will be hopefully, as soon as possible, the clinical trial. But this is going to require competencies and skills we don't have as a so small company of 2 employees. So we are also now actively looking for a partner as a pharmaceutical company to co-develop this molecule or to out-license it. So this is what I wanted to present today, and I thank you for your attention.
Any questions for Dr. Demotz? We have a few minutes for questions.
How can we get blood to you to be able to be tested to see if our children would be viable for the study?
I don't know if today this would be useful. In the sense, it was more when we went through what Amicus Therapeutics did during the development of their molecule. They had a hard time with the identification of the right patient. So we learned from their problems. So we started to develop this assay earlier than they have done. So it seems that it goes in the right direction. So we wanted to avoid the situation to be close to the clinical trial and not to have the right tool to identify the right patients. It's not the thing to get more samples is not going to make it faster for us. We have there are other.
How do I, as a parent, have hope in this? Like, how do I know that my child could potentially be, a part of this study? That's what interests me.
Okay. So you probably know the mutations of your child, and since we have now tested quite a few, it's probably there is a good likelihood.
Is there a list available of those mutations that fall within the feasibility?
we have not. It's not published, but we can discuss.
Yesterday they talked about an issue they had bumped into with testing larger animals or animals with larger brains, like such as a monkey, and they had issues with that. Is that something that you guys are going to try as well, or is that not going to be an issue with this kind of therapy?
We want to test the how to say that. People would be reluctant to go to start a clinical trial without the testing of the compound in an animal model. I'm not personally convinced that it was going to bring much, but we are going to do it. So there are at least 3 animal models available. There is a mouse, there is a cat, and we'll hear about the cat models very soon. And there is a sheep model. So we are in discussion with the 3 animals.
As I understand mechanism of chaperones, if you don't give enough, it may not work. If you give too much, it may work as an inhibitor. And my understanding, the dosage is a very, let me finish, the dosage is a very difficult problem, and I want to find out how you're going to determine dosage and test patient for a response. What type of testing you're going to get, okay, to see if it's responding?
It's not an inhibitor even at high concentration. Surprisingly, this molecule is a very potent inhibitor at pH 7 in the environment of the endoplasmic reticulum, and it's no longer it gets protonated at the acidic pH of the lysosomes. It's no longer an inhibitor. And when you do the chaperone assay in vitro, it reaches a plateau, and we have not been able to reach a concentration where there is inhibition. And this may be with gram per ml, but we're never going to reach such concentrations. So it seems, at least in vitro, there is no inhibition observed.
But still, how are you going to determine what is an effective dose? How are you going to test a patient what their response? Are you going to work with biomarkers as since Dr. Tiff described? Is there anything a little bit more precise? You cannot do MRI every day, so is it any type of a back loop mechanism for testing of your compound?
But to determine the dose we have started to do this in mice by doing PK in the brain PK. So now we start to have an idea of how much compound has to be administered in mice at least. Then it has to be done in higher species before to go in humans. This is a monkey, probably a brain PK in monkey as a last step. And then the biomarkers, this is, yes, this is in discussion.
Okay.
Yeah, I noticed on your slide that you work with Totori University.
Yes.
Some of us parents have sent blood samples to Dr. Higaki about almost a similar finding. Are you working with him in terms of, or is this a very different study that you're doing?
No, we are working, he has tested this compound on some cells he has that we didn't have. Yes, and we are discussing about running this mouse study with him.
Okay, so it's the same compound, or the same?
No, there are several compounds. There are 2 Japanese compounds and slash Spanish, but they are not, to our knowledge, they have problem of synthesis and there is toxicity because it's not. But we can discuss this for the sake of time.
Okay. Yeah,
if it's okay.
In the reception, the 2 of you offline. Do you have a quick one?
Yes. A quick one.
Okay, sorry.
Real quick. Is this a compound like a similar, or it's a available drug, FDA approved? We can try the compassion use? No.
No, it's not.
Okay, thank you very much again. That was fascinating.